Well, welcome back again to our narrated presentations for the chapters associated with this class. This is our second narrated presentation, and today we are looking at the evolution of populations, what is sometimes referred to as population genetics. And that actually leads us to one of the things that is most misunderstood about the whole process of evolution, the fact that individuals do not evolve. And that's something that I touched on at the end of the last presentation, but it's something that we're going to reinforce with this presentation. The fact that when you are looking for evolutionary changes, you cannot look at individuals because individuals do not change, they do not evolve, or at least not in the way that is relevant as far as the evolutionary process. So you may evolve culturally, you may evolve as an individual over the course of your lifetime, but those changes are changes that will only be unique to you. And unless they are encoded into the DNA that you pass on to your offspring, they are going to die with you when you die out. And so it is one of the tenets of evolutionary theory that individuals are not the units of evolution. So what does constitute the unit of evolution if individuals don't represent the unit of evolution, because we don't actually observe individuals evolving? Well, the answer can be seen by taking a look at where Darwin and Wallace observed genetic variation, because it is genetic variation that is the raw material that natural selection, the engine of evolution, is going to operate. And when we start looking for genetic variation, we observe genetic variation in individuals, but those individuals are part of a population. So genetic variation, the raw material for natural selection, is observed in populations as a consequence of differences amongst individuals. So while individuals are not going to evolve, the populations in which they exist and the individuals from generation to generation certainly do have the opportunity to evolve, not in the sense of individuals evolving, but the makeup of the overall population, the genetics and the phenotypes of the individuals that are present in each population from one generation to the next. That can evolve and that does change. And that was one of the initial observations that both Wallace and Darwin made, leading to their development of the concept of natural selection. The fact that individuals showed variation within a population, and it's populations that show these variations and changes over time in these variations. So the smallest unit of evolution is a population. That's what we see evolving. And so that's what we're going to look at today. How can we document changes in populations over time? And there are ways of doing this. And what's the significance of that? What can we tell about the changes in population makeup over time that tells us that a population is evolving? And is natural selection the only way in which a population can evolve? And in simply asking that question, you should realize that the answer is there are other ways in which populations evolve, but natural selection is the only one that leads to adaptation of populations over time. And that's very important to understand as well. So the first thing to remember is that individuals show certain features, and those features belong to them for a lifetime because their genetics is fixed at the point of conception, when egg meets sperm for asexually reproducing organisms. And that genetic makeup is not going to change significantly over the course of the lifetime of the organism. Yes, they may accumulate mutations, and we will talk about the role of mutations very shortly. they may accumulate mutations which may have an effect on the overall lifespan of the organism. But unless those mutations occur in the germ cells that are going to give rise to eggs and sperm, those mutations will exist only for the course of the lifetime of that organism and will die with that organism. But we can also see changes in features over time. And what you're seeing here are some of the Darwinian finches of the Galapagos, and this is based on the work of Peter and Rosemary Grant, who I introduced you to very, very quickly in the last presentation. As I mentioned then, they have been documenting the physical features, the phenotypes of finches on a small island in the Galapagos for the best part of 30 years now. And they have consistently been able to document changes in beak size and other phenotypes as a consequence of environmental changes, usually in association with drought. And as I mentioned at the end of the last presentation, they've also in the last 15 years or so now been able to document the emergence of a brand new species of finch on this island as a consequence of an immigrant who they named Big Boy because he was larger than the males of the two species that occupied the island where they're working, who turned up on the island some years ago, found some females that he was able to mate with, and Big Boy and his offspring constitute a new species because they are now incapable of backbreeding with either of the two existing species that have previously occupied that island. So Big Boy and his offspring being reproductively isolated, and we'll talk about the significance of that in an upcoming presentation, now represent a new species that is emerging. So there are now 3 species occupying this island, the 2 species that were originally there when the Grants arrived, and this new species that has turned up as a consequence of Big Boy. But I digress a little bit. What the Grants have documented is that physical features of the 2 existing species of finches on the island, and I assume Big Boy and his group as well, change with environmental changes. And it's very easy to document this through the very close physical measurements that the Grants made over the course of the last 30 years. And so we know that individuals do show particular features. There is variation amongst individuals in populations, and those physical features can change from one generation to the next in response to environmental changes. This is easy to document. So the question then becomes, why do these changes occur? And those changes occur because the underlying frequency of the alleles that are present in the population change with time. And this is where the discipline of population genetics comes in. Population genetics studies how populations change over time and specifically focus on changes in allele frequencies amongst the populations. What we refer to today as the gene pool. The gene pool is identified as the combination of all the alleles in all the individuals in a population. And we'll come back and we'll talk about gene pools again a little bit later in this first part of the presentation, right at the end before we take a break. But this represents a coming together of the Darwin-Wallace theory of evolution and Mendel's observations of inheritance, the 2 principles that he had in place. These came together very early in the 20th century, shortly after people recognized the significance of Mendel's ideas. And it was kind of important that they did come together because both Wallace and Darwin knew that variation existed in populations. They just couldn't explain where it came from. They knew it had to be something to do with what was happening from one generation to the next, but they had no mechanism in place to explain the inheritance of characteristics from one generation to the next. Ironically, Mendel, who was a contemporary of Darwin, so somewhat of a contemporary of Darwin's, living no more than perhaps 300-400 miles from where Darwin was located, had the answer. And while Mendel was familiar with Darwin's work, we know he had a copy of On the Origin of Species, and he certainly read it because he made many notations in the margins of the book. Darwin was apparently not aware of Mendel's work. There is some suspicion that maybe he was, but there is no direct evidence to support the idea that Darwin had a familiarity with Mendel's work. And certainly there does not appear to have been any form of communication between Darwin and Mendel. So while both Darwin and Wallace were perfectly aware of these variations in populations, they couldn't explain the origin of these variations. Mendel could, through his observations of inheritance in his garden piece. And so once Mendel's work was recognized for its significance, it very quickly became obvious that you had to blend Darwin and Wallace and Mendel together. And this is the origin of population genetics, this bringing together of Darwin's and Wallace's ideas with Mendel's observations of inheritance. And so this is often referred to as the modern synthesis. You may actually have heard that term previously in a Biology 1 course. We sometimes introduce you to the notion of the modern synthesis. But here we want to build on this idea of the integration of Mendelian genetics and Darwinian or Darwinian Wallace evolution, and take a look at this idea of populations as the unit of evolution. So the first thing to recognize is exactly where variation comes from in any population. And there are two sources of variation. The first is mutation, and mutation is very, very significant because, as you may have learned in Biology 1, but which you must very much be aware of now, is that mutation is the only source of new alleles. Because remember, an allele is a variation or a flavor of a gene, and it represents a change in the nucleotide sequence in that region of DNA that represents the gene. So as soon as you change a single nucleotide in a gene sequence, you've created a new allele. But of course, changing a nucleotide in a gene sequence is a mutation. So mutations are the only source of new alleles. So the significance of mutations cannot be overlooked. We have to recognize that mutations are really important because they are the only source of alleles that lead to genetic variation. So once we recognize that, the next thing to recognize is that the vast majority of mutations never get transmitted into the broader population. They appear in an individual and they disappear when that individual dies. Why? Because unfortunately, many mutations are lethal. And if they're not lethal, the vast majority are neutral. They have no adaptive impact at all. What we also need to recognize is that the only mutations that are going to count as far as generational significance is going to be concerned are those that are occurring in the germ cells. And those are very limited in number. The vast majority of mutations are going to occur in somatic cells. The somatic cells will never make a contribution to the next generation. So any mutations that occur in somatic cells, they are of no consequence. We don't even have to worry about those, other than the effect that they will have on the individual that carries them. But in terms of evolution and natural selection, they don't count because they're never going to be passed on to the next generation. So only mutations that occur in germ cells that have any significance, and only a very limited number of those are going to have an impact on the population in which they occur. Because unless that mutation creates a phenotype that is timely, has relevance at that particular time point, and also has geographic significance. In other words, it also matches the geographic location in which it appears. Again, those mutations are going to have no significance. So while mutations are really, really important because they are the source of new alleles, unless they occur in germ cells and unless they occur at the right time and in the right location, they're not going to have any significance, but we still can't rule out the significance of mutations. We can't ignore the significance of mutations. We do know too that the rates of mutation vary from organism to organism. So in organisms that are diploid and are typically multicellular, the impact of mutations is relatively low. And the rate of mutations that are of significance is very, very low in multicellular organisms. But in organisms that are unicellular, and particularly organisms that have a single copy of each gene, in other words, what we might refer to as functionally haploid, whether they be eukaryotic or prokaryotic, any mutation that occurs in the DNA is going to be of significance, because given that there is only a single copy of every gene, if that gene gets mutated, there is the potential for very significant changes, because now you've changed the single copy of that gene. And particularly if the mutation has significance, whether it be lethal or whether it be beneficial, that's going to affect those microorganisms, particularly if it can be distributed on into the more general population. Now, for asexually reproducing organisms, those are only going to get transmitted into the population if they are significant and they allow the one individual in which the mutation has occurred to have a advantage over cells nearby. The individual then will divide into two, those two cells will divide into four, the benefit of compounding. And all of those cells, because they are genetically identical, bearing the same advantageous mutation, will come to dominate the landscape. In the case of sexually reproducing organisms, that situation may be a little bit more complicated. Again, it depends on the nature of the mutation. Is it occurring in a cell that is going to give rise to gametes? And is it going to be of significance? If it's in the somatic cells, as I mentioned earlier, it's not going to have any benefit at all. It's not going to be passed on to next generations. So when we start thinking in terms of mutation rates, we also need to recognize that it varies from organism to organism and types of organism. There is no uniform way of measuring mutation rates. They vary. We also need to recognize that while we do see genetic variation, not all genetic variation is heritable. So even if we do see some form of variation in a population, we can't automatically assume that it is heritable and therefore going to be passed on to subsequent generations. And here's a really great example. The book talks about snakes, but the example that I've got from a previous edition of the textbook, I think, is even more dramatic. Because here are some caterpillars from the same species of butterfly, if I recall correctly. I think these are caterpillars for a butterfly. And while you may look at these caterpillars and go, surely they're from different species, they are so phenotypically different. The reason why they're different is not because of their genetics. Genetically, they're identical, and that can be demonstrated very easily. What is making the difference is the chemicals in the food sources that they're feeding from. This is a feature of a relatively new discipline in biology, which hopefully you encountered in your Biology 1 course, called epigenomics, where the environment impacts gene expression. Not only does gene expression impact interactions with the environment actually feeds back on gene expression. And this is a great example of that. So the chemicals that the caterpillars on the left are obtaining from feeding on leaves interact with the genes to give rise to this particular phenotype that you can see, which looks very much like a flower bud arrangement. In the caterpillar on the right, feeding on the twig material, it's getting exposed to a different set of chemicals, and those chemicals interacting with the same set of genes give rise to a different phenotype that looks very much like a twig. However, this is in response not to difference in genetics. They are genetically identical, but in terms of the environmental exposure that they're getting, specifically the chemicals in their food, giving rise to these two phenotypes. So if you were to transfer these caterpillars between the two environments, you could expect their phenotypes to change. Or if the resulting butterflies that would emerge from these two caterpillars, two different types of caterpillars, were to lay their eggs in the opposing environment, their caterpillars would emerge and develop in response to the environment that they're in, not the environment that their parents were raised in, because these are not inheritable traits. So we should not automatically assume that just because we see changes or we see differences in populations that these are necessarily going to be inherited, because they don't have to be. This discipline of epigenomics is showing very clearly the role that environment plays in the type of phenotypes that individuals express. And if you've ever been around a pair of identical twins, you've experienced this. Or if you are a member of an identical twin pair, you have almost certainly experienced this, because twins do not experience their environment identically, and there are subtle changes that emerge as twins get older. This is, again, epigenomics. So even though they share identical genetics, the impact of the environment changes the phenotypes sometimes in very subtle ways, other times not so much. Sometimes it's a little bit more obvious, as you see here. So when we come to thinking in terms of genetic variation in populations, we measure that variation in the terms of something called polymorphisms. Polymorphisms can exist at two different levels, and we'll look at those two different levels shortly. But what is important is to get a measure of what is called heterozygosity. Heterozygosity refers to what you might imagine it refers to. If you think in terms of genotypes for organisms, you know that there's the homozygous genotype, either homozygous dominant or recessive, and there's heterozygous, where the alleles are different on the two homologous chromosomes, thinking back to bio 1. So what we're looking for here is how many of the loci, the physical locations of genes on chromosomes, contain heterozygous alleles. In other words, the two alleles at these particular positions are not identical for the two copies of the same gene. They're actually two different alleles. This is referred to as the level of heterozygosity, and what we're looking for is the average heterozygosity in a particular population. But remember, I said polymorphisms can occur in two different types. We measure heterozygosity at the gene level. So there are genetic polymorphisms that we can recognize, but there are also physical polymorphisms. And they turn up in populations in different ways. But before I discuss the two different types of polymorphisms a little bit more closely, just so you have a better feel for what they look like, when we want to measure genetic variability or heterozygosity, we do so by looking at DNA sequences. And we can look at DNA sequences, and we look for changes in DNA sequences by looking for base pair substitutions, insertions, and deletions, and comparing sequences between related species. We'll look more at the examination of genetic sequences in a later chapter, when we start to look at how we can use DNA sequencing as a way of determining relationship between organisms. But for now, all we need to recognize is that mutations give rise to alleles, and we can look at heterozygosity by measuring the number of substitutions that have occurred, the number of insertions and deletions that have occurred, and what effect those have on the amino acid sequences of the resulting polypeptides, because it's really at the polypeptide polypeptide level, that we see the effect of the mutations and the potential effect on phenotypes. So if we have two alleles where the substitutions, particularly the substitutions, don't alter the amino acid sequence, what we refer to as a silent mutation, even though we would recognize those as two separate alleles, in terms of their effect on variation, they're not going to have any effect on variation because, at least at the phenotype level, because the two resulting polypeptides are going to be exactly the same. So we do also have to differentiate between the types of mutations that exist in order to establish the nature of the heterozygosity and whether it's going to have an effect over time on the population. So it comes down to looking at DNA sequences. So there are other changes that can also occur at the chromosome level. You may find changes that result in gross changes to chromosome structure. And you should have been introduced to some of those again in your Biology 1 course, where you have disruption in the exchange of DNA between the homologous pairs of chromosomes during crossing over. So the exchange is supposed to be reciprocal. A fragment from each of the two non-sister chromatids gets exchanged. So the movement is in both directions. It could, however, be potentially unidirectional, which would lead to a duplication in one chromosome and a deletion in the partner homologue. You could have an inversion where there is the exchange of the fragment, but it is inserted into the recipient chromatid in the reverse orientation. There are a number of different gross changes that can occur in the chromosome as a result of incorrect crossing over, I guess, or incorrect exchange during crossing over. So we also need to take that into account. There are a number of different ways in which changes to chromosomes can occur. Then to distribute the resulting alleles that are beneficial, we need sexual reproduction. Why do we need sexual reproduction? Because it is in meiosis and the behavior of the chromosomes in meiosis that we see the possibility for creating new combinations of alleles and new combinations of genes as a consequence of the crossing over process, the random alignment of chromosomes on the metaphase plate in meiosis 1 that leads to all possible combinations of chromosomes in the gametes, Mendel's second law of independent assortment. Then we have the random coming together of the gametes, egg and sperm, and all the possible combinations that are there. So for humans, there are 2 to the power 23 possible combinations of chromosomes in the gametes. And then when 2 individuals produce offspring, each of them bring those 2 to the 23 possible combinations, which gives a possible number of offspring in the order of 2 to the power 23 squared, which is 2 to the power 46, which is somewhere north of, what's the number? 3 trillion, if I remember correctly. I need to go back and take a look at the number. This is something that you should hopefully have been introduced to again in your Biology 1 class. With all of those possibilities, and then you have to remember that crossing over itself creates essentially an infinite number of possibilities, because we do not know where or when or if crossing over is going to occur between any pair of homologous chromosomes. We basically have a huge opportunity for genetic variation. So sexual reproduction is also incredibly important for the distribution of a beneficial allele from the individual in which it first emerges into a wider percentage of the population. It's not going to happen in a similar or in anywhere like the same percentage-wise distribution into subsequent generations in terms of retention of the favorable allele over the long term in organisms that are reproducing asexually. Because remember again, in asexual reproduction, the offspring are clones of each other, they're genetically identical, and any environmental change that affects one individual is going to affect all of them. Whereas with sexual reproduction and this possibility for large amounts of genetic variation, just because there is a change in the environment doesn't mean that everybody's going to get affected equally. So sexual reproduction provides that opportunity for the distribution of favorable alleles into a wider percentage of the population. So when we look at the variation within a population, we recognize that there are two different types of variation. There's discrete variation. This would be Mendel's purple versus white flowers. So these are the either or, where there is one form or another form and no variation in between, no blending of characteristics to give alternative variations. And then there is what is referred to as a quantitative character, where you see a continuum of variation in the population. So a couple of examples that hopefully you learnt about in Biology 1 again is something like human height, where there is a continuum of height in human populations varying from around 4'9", which is at the lowest end of normal human height, to around 6'4", which is considered to be the the other extreme of normal human height. Dwarfism and gigantism, extremely short and extremely tall, lie outside of the continuum of normal human height. Skin color is another one that is often talked about here. And again, we don't include albinism in this because albinism is a product of its own mutation. And we do not think in terms of an extreme at the other end of the axis other than one that is part of the continuum, because skin color is a multi-genetic characteristic, and each of the genes involved has multiple alleles. But that is the characteristic of a quantitative character. A quantitative character is not the product of a single gene with a limited number of alleles. In Mendel's case, we would say 2 alleles, which would give the either or characteristics. In quantitative characters, there are multiple genes involved, and typically there are at least 2 alleles for each gene, maybe more alleles for each gene. But that's what gives rise to the continuum. When we take a look then at the two different types of polymorphisms, we do recognize that these polymorphisms also, in some cases, give rise to genetic polymorphisms. So when you have a phenotypic polymorphism, what you're really referring to is phenotypes that are of high enough frequency in the population that you actually recognize that they're there. So if you had a single individual in a population showing a unique phenotype, that may not be representative of a polymorphic population, because you've got a single individual. You would need more individuals showing the same phenotype for the population to be considered to be phenotypically polymorph. However, at the genetic level, if you know that you've got a quantitative character, one that shows that continuum of phenotypes, then you can be very certain that you've got a genotypic polymorphism, where there are multiple heritable combinations of genes that give rise to the characters. And those characters show that continuum of features in the population. And so genetic polymorphisms and phenotypic polymorphisms are the two ways in which we can identify the presence of genetic variation. Phenotypic polymorphism typically is associated with discrete characters, the either or situation. Genetic polymorphisms tend to be associated with quantitative characters where there is a continuum of phenotypes within the population. So we have two different types of polymorphisms that we can look at. Both of them allow us to get a measure of genetic variation in a population. So when we start thinking in terms of variations amongst populations, there are two ways in which we can look at variations amongst populations. What we've been looking at right now is variation within a population, but we can also see variation between two populations, and they don't necessarily have to be strictly geographically isolated from each other, but in most cases where we do see genetic variation between populations, there is a level of geographic isolation. So let's look at some examples of this. So here's an example of genetic variation in a population, and this is at the gross chromosomal level. So these are two populations of mice that are found on the island of Madeira. This was settled by Portuguese colonists back in the 1500s. So we know about when the mice arrived, because the mice arrived with the colonists. And so we see these two discrete populations that still have some connection to each other. But in terms of the chromosomal makeup of the two mice, while they are genetically identical, meaning they have the same set of genes, those genes are now located on very distinctly different chromosomes. And where you see two numbers separated by a dot, like 2.4 or 13.4 or 13.17, what you're looking at is a chromosomal fusion between those two chromosomes from the original populations of mice that colonized this island. And you can see that many of the chromosomes have undergone chromosomal fusion, reducing the number of chromosomes. But in these two populations, the type of fusions that have occurred are very, very different. In some cases, they share the same fusion product, which suggests that that fusion occurred before these two populations segregated, while they still represented a single population. But in other cases, you could see quite clearly that they have their own unique chromosomal fusions. And this suggests very strongly that these two populations are actually on the way to becoming separate species. And at some point in the future, they may actually be identified as separate species. But for now, they still are considered to be subspecies of the same species. But this separation is occurring because of these gross chromosomal changes as a consequence of chromosomal fusion. So phenotypically, these mice do not look dissimilar from each other. They look very similar to each other, but they are chromosomally separate from each other, distinct from each other. And so there's a level of genetic variation between these two populations at the gross chromosomal level. Then in some circumstances, we find geographic variations between a species that is not divided up into geographically isolated populations, but consists of a single continuous group of populations that are interacting with each other and overlapping in terms of their geographic range. This is what we refer to as a cline, a geographic cline. So here is a diagram of a genetic cline as a response to a geographical cline. This is a species of fish that lives along the east coast of the United States, from Georgia up into Maine. Now, the geographic cline that's present here is the change in water temperature. So clearly in Georgia, you have the influence of the Gulf Stream, you have a generally warmer climate, so you would expect the ocean waters to be a lot warmer. But as you move northwards, the effect of the Gulf Stream as it moves away from the continental United States and starts its transition across the Atlantic Ocean, leaves behind cooler water. And of course, as you move further north, it gets cooler as well. And you would expect that a fish that originates in warm water would probably not do terribly well in cold water. But this particular species of fish has a broad geographic range, and it is able to tolerate the cooler waters as you move northwards. And there is a good explanation for why this is the case. There is a mutation of the gene LDH, which is lactate dehydrogenase. And lactate dehydrogenase is a central gene in cellular respiration. And you would expect that a warm water fish would have a very good level of cellular respiration because it is warmer. And if it got into cooler water, cellular respiration would not do quite so well because fish cannot maintain a warm internal body temperature. But the fish that of this species that live in the northern latitudes have this mutation of the lactate dehydrogenase B gene. So it's the subunit B or beta of the lactate dehydrogenase enzyme that is more efficient in cooler temperatures. It's actually an adaptation to cooler temperatures. And you see this reflected in the genetic makeup of the populations. So as you move further north, because of the overlap of the regional populations along the east coast of the continental US, there is a movement of alleles from one population to the next because they can interbreed. And so you see first the emergence of the cold water allele that allows the fish to migrate further north and take advantage of new opportunities in the form of new environments. But because the populations are not isolated from each other, they overlap, and therefore there is interbreeding between the populations. You see the transfer of the allele between these overlapping populations. In the higher latitude populations, this allele is almost fixed in the population. It's the only version of the allele that's present. As you move further south, you see an increasing frequency of the warm water version of the allele, so that by the time you get into the warm water latitudes further south, you see very low frequencies of the cold water mutation and almost exclusively the warm water mutation. This is what we see in a geographical climate where the populations exist as overlapping populations rather than discrete populations. But even in situations where populations overlap, you may not see a lot of movement of alleles between populations because the two populations may remain more or less discrete from each other with very little interbreeding. So a good example of this is these two reindeer herds in Alaska and Canada. So you've got the porcupine herd that lives on the plains close to the ocean, so on the flatlands closer to the ocean. And then you've got the 40-mile herd range that lives further inland and occupies region in the foothills of the mountain ranges that dominate this regions of Alaska and Canada. You can see that at times the ranges of the two herds do overlap. So there's at least the potential for interbreeding between the two herds. But genetic analysis suggests that there is very little exchange between the two herds. And this is because the males of the two herds compete with other males of the two herds for maintenance of harems of females. And the males tend to maintain their harems amongst the females of their own herds, with very little opportunity for females from the opposing herd to enter into a harem in the other herd. And so there's very little opportunity for breeding between the two herds because of the nature of the breeding arrangements of these two caribou herds. And so even though there is an overlap between these two populations, that does not imply that there's going to be an ongoing exchange of genes and alleles between the two herds, because unless there is an ongoing opportunity for significant amounts of interbreeding, it's simply not going to happen. So in this case, even though we could argue that there's the potential for a geographic line, one does not exist because there's no interbreeding between the overlapping populations, or no significant amount of interbreeding between the overlapping populations. So let's stop for a few minutes, take a break. This is a good opportunity to take one. And as always, I've got some concept check questions that I will answer when we come back from the break. So here are the two questions. Why is genetic variation in a population a requirement for evolution? Hopefully that's pretty obvious, but we'll see. And mutations occur spontaneously in populations all the time. You can't eliminate the possibility of mutations. So why do only a small number become widespread in a population? We've actually discussed the answers to these two questions in this previous section. But I'll see you on the other side of the break with the answers.